From Microbe TV, this is a special immune booster recorded during the American Association of Immunologists conference in Chicago, May 2024, starring Cindy Leifer and Steph Langle, and featuring David Martinez from the Yale Institute for Global Health.
Tell us when you first got excited about science, and who were your mentors and people who really inspired you back then?
So I think in my case, perhaps it's a little bit boring in that I have the cliche, as far back as I can remember, I was always interested in science. So I think right around the age of 4 or 5 years old, I had my father was a physician, and he had laboratory notebooks in his clinic. So, of course, I wasn't doing experiments, but I was not at the time when I was 4, although later I was, trying to do my own experiments. But I was really interested in animals, like many little children are. And I used to collect these cutouts of animals in their little descriptions, and I used to paste them into this laboratory notebook so I could reference it and sort of go back to it. So by no means it is a scientific experiment, but certainly an inclination towards science from an early age.
Sure. So it does sound like your dad, though, being a physician, but also having laboratory notebooks. He was a detail-oriented person. He was thinking in this experimental type of way.
Correct. So I grew up in El Salvador, and El Salvador doesn't have a strong infrastructure for biomedical research. So I think as far as I know, I don't think the country has a single institution that can award a PhD in biomedical sciences. Now, they have other doctoral programs for, business-like areas, but as far as biomedical science, and I could be wrong, but I'm pretty sure they do not have one. So you're correct in that, he didn't have a formal scientific training, but certainly was interested in, biomedical science from an enthusiasm point of view.
Right. That's great. And so as you then are moving towards, your love of science because of your father and the supportive, I'm assuming probably educators helped you kind of get to where you were in grad school. So can you talk about that transition from, you're interested in science, but did you know you could have a career in science, a career as a scientist?
I did not know I could have a career in science until probably earlier than most people, actually. But when I was a senior in high school, I worked in the summer between my senior year of high school and freshman year of college. I worked at this institute called the Nobel Foundation, and that is a plant biology research institute in my hometown in the United States, at least, in Ardmore, Oklahoma, of all places. So, they focus on, research related to agriculture, including, making, better alfalfa so that, cattle can, feed on the alfalfa and not get bloat, for example, among many other things they focus on. So more of an applied type of agricultural research setting. But at the time, I had a neighbor who, in fact, I met up with him this last summer. He came to my lab and visited, but he was a scientist and he lived in my neighborhood. And I, ran into him one time right after I had graduated, and he mentioned that I could perhaps work at the Nobel Foundation. And then he hooked me up with, someone who he thought would be good for my career. It was this scientist of really high stature, who had been elected into the National Academy of Sciences. In plant biology, that's more rare, just because of it's clearly an important area, but certainly gets less notoriety than it should, in my opinion. But his name is Dr. Richard Dixon, the lab of the scientist who I worked in, and just through interacting with him, he was really an inspirational figure in seeing how science was done, and then seeing that one could be a scientist and also be a highly successful scientist. So that, being around Dr. Dixon and working in his lab was really inspiring for me as I began college.
And did you continue research once you got into college?
I did. So I joined a lab as a sophomore, a bacteriology lab at the University of Oklahoma. So, from day one, I was a microbiology major. I never changed my major. And because I was, just fascinated by tiny little creatures and how they could, manipulate, cells to essentially copy themselves. So I did join the laboratory of Dr. Paul Lawson at the University of Oklahoma. And then as a senior, I took a advanced topics in virology course. And that's when I became exposed to viruses in a more formal setting. And I decided to apply to, the SURF programs, these summer undergraduate research programs. And I was actually near to where we are here in Chicago. I was a few hours north. I got into a program at the University of Wisconsin-Madison. And I worked with Rob Kaleda on studying cytomegalovirus pathogenesis and understanding the role of certain tegument proteins in being able to reactivate the virus from a latency setting. So that was sort of my first real exposure to virology in a more formal setting. And that's when I got hooked on viruses and host pathogen interactions.
Awesome. Well, it's interesting that you talked about little critters that you put in your notebook when you were 4, and then you're now thinking about little critters under the microscope as viruses.
Yeah,
So.
and we still record information about those critters in a notebook.
Exactly.
And it's a circle.
Comes full circle, yeah. Yeah.
Well, that's great. So at this point, okay, scientist is kind of for me. You then go to grad school. So tell us about your graduate training and then kind of what led after.
Sure. So, I think perhaps naively I applied to, I was really interested in HIV whenever I took this advanced, topics in virology course. And I was interested in HIV because I was perplexed by the fact that HIV at the time had been studied for over 3 decades, and still we had no HIV vaccine. So I was just fascinated by that concept as to why that was. And I decided to apply to programs that I thought were really strong at HIV immunity. And I, essentially ended up choosing what I thought was the best program to study HIV immunity. And that led me to Duke University. And at the time, Duke University, and I think still, they're home to the Duke Human Vaccine Institute, which is actually where Steph and I interfaced initially. And I was just interested in rotating in various labs there. And I rotated with Sally Permar as my first rotation. And I, perhaps should have done additional rotations, but, it turned out really well. But I decided I wanted to join that lab. So I went and did a second rotation. But after that, I went back and joined Sally's.
Oh, how cool. So they didn't even make you do that third one.
They didn't make me do a
sure.
third one. I told them, I said, I'm sure about this. I'm going back to Sally's lab. And I think they must have believed me that I was sure because they didn't oppose anything.
Yeah.
So you guys overlapped, right? Or not?
I don't actually think so.
We didn't overlap, but I was still in the lab when you interviewed.
When I interviewed. Yes, I do remember that. And we
okay.
talked about your science and you were kind of on your way to your postdoc and I was coming for my postdoc. So
Correct. Yeah. So we had a slight overlap.
Yeah.
And at least connecting.
Yeah. Great.
And then did you, so you moved with Sally, so you were already done in Sally's lab.
Well, so
then?
Sally we can talk a little bit about Sally's career trajectory, but Sally went to Weill Cornell
right.
in the middle of when I was a postdoc with her. I did not want to move to New York City.
Oh, right.
This
so
time,
you stayed.
So I stayed.
Yeah.
But at this time, David had graduated with his PhD.
Mm-hmm.
And where did you go after
Yeah,
that? Which
where
was
did you go?
relevant to 2020.
Right. So I went to Ralph Barrick's lab at UNC Chapel Hill.
Mm-hmm.
And there's really multiple reasons for that. So, one of them was very practical, is that my wife was finishing her PhD at UNC Chapel Hill, and she was, 2 years behind me. So I thought, I initially thought maybe we could live apart, but then I thought about it again and I didn't want to move away. But really, perhaps more importantly was that in 2015, when I was, I think, a rising third-year grad student, was when the Zika virus epidemic really blew up. And as I took this advanced course in, virology as an undergraduate, I had learned about, sort of the concept of viral emergence, looking back to, 1976 with Marburg emerging, or at least first being detected and recognized as an emerging filovirus. And then in the early 1980s, you had the emergence of HIV at the time called HTLV, the virus that causes AIDS. Then fast forwarding, a few years later, early 2000s, the emergence of SARS-CoV, what most people call SARS-CoV-1. And again, not to, beat a dead horse, but, you keep fast forwarding, 2011, 2012, MERS coronavirus emerges. And then, Ebola right after that in 2014, and then Zika. So I thought I should go to a lab that is really good at studying emerging viruses. And that led me to the Barrick lab. So I interviewed in Ralph's lab and was, thankfully he had a slot for me. And it was really sort of an example of being at the right place at the right time.
You definitely were astute in thinking about, if you want to work in emerging infectious diseases, working with somebody like that who's doing that work, and from what at least I could see from the outside, seems to be very supportive of his people.
He's a
and
fantastic mentor.
Comment on that, yeah.
Yeah. So Ralph is absolutely wonderful as a mentor. In terms of letting you explore ideas you're interested in. Thankfully at the time, and my understanding is that even to date, he's thankfully well funded, so he has the capacity to kind of let people explore things that he's also interested in, but perhaps hasn't thought about yet. So he was, really sort of instrumental in letting me, play in the lab in terms of, pursuing ideas that I was interested in, which is something that I hope to emulate.
So what were some of those things that you were studying in that lab at the time?
Yeah. So, I was initially, both sort of, I had both feet in, the flavivirus out of the lab as well as the coronavirus out of the lab. And to be 100% honest with you, at the time before COVID had emerged, I wasn't sure if my program had a future with coronaviruses, primarily because they had been so difficult to fund, and I was aware of this. Yes. So it wasn't that I thought they weren't important. I clearly thought they were. But the sentiment in the field was that, in the emerging infectious disease field, was that, coronaviruses are not relevant, and I even, remember, obviously won't mention any names, but, in grad school, I had a graduate student at Duke University tell me that he didn't think we needed to have vaccines for emerging coronaviruses. So I thought, well, that's a really interesting, that's an interesting idea. And in hindsight, hindsight can be 20/20.
Yeah,
exactly.
You're like, I wish we would have put a bet on that.
I wish I would have placed a bet. Yeah. So I was initially interested in, looking at how different dengue virus serotypes, how they vary genetically and what the impact of that genetic variation had on the immune response. Mm-hmm. So, this idea was perhaps, borrowed from the HIV field, in that, HIV is sort of the paragon of genetic diversity within RNA viruses. And, I thought maybe if we can try to understand what the role of genetic diversity is within the serotypes, we might learn something new. And it turns out that, the role of genetic variation within each serotype actually majorly impacts the neutralizing antibody response evasion. So that was initially what I did in Ralph's lab. And right around the time when COVID emerged, I had just finished my BSL-3 training. So that was really
convenient.
Opportune and convenient timing.
Yeah. And so your time, you joined that lab and SARS-CoV-2 emerges. And so then you are probably, I'm imagining, in the lab. I mean, I don't know. What was your schedule like? I mean, it was probably a wild time to be a part of that. It
was a wild time. So we worked very hard. A lot of people in the lab worked very hard. It certainly wasn't a 24/7 thing. If you worked in a BSL-3, that
demanding,
it's
right?
very demanding. It's very physically demanding as well. You can't go to the bathroom if you're in there. So you would have to, degown and, answer nature's calling and then, come back. But, the BSCs were in high demand. So the biological safety cabinets. So you really, time your schedule around the availability of those. So it was really demanding, but I think early on, and, this is when my training in HIV, I guess, became relevant, is that as an HIV, as a student studying the immune response to HIV, sort of in a vertical transmission setting, I utilized this luciferase HIV neutralization assay. And I knew that in the Barrick lab, we had a variety of coronaviruses that express luciferase. And of course, the idea was then to very early in the pandemic, even before we knew how what the magnitude of this thing would be. My idea was to develop a high throughput neutralization assay for SARS-CoV-2. Now, this was even before we had, fully recovered the clone that expressed an antiluciferase virus. So I was actually optimizing a neutralizing antibody assay for MERS and SARS because we had the nanoluciferase viruses for that. So, basically optimize that so that for those two viruses, so that by the time we were able to recover the SARS-CoV-2 nanoluciferase assay, I had all the conditions optimized and essentially was able to get that up and running very quickly. And I think I might have even said to Ralph, people are going to be lining up, at the door for this reagent.
Absolutely.
Yeah.
And that turned out to be the case.
You're like, that was correct. Yes.
So what kinds of things were you able to do with that? What were your major discoveries with that assay and around those kinds of experiments?
Sure. So,
SARS-CoV-2.
So that sort of evolved over time. So, initially, we were primarily collaborating with various people outside of UNC. So I think the first use of those nanoluciferase viruses was to show the immune response that was generated from macaques that were initially infected and then re-challenged. So what we were doing in collaboration with Dan Baruch at Harvard was to evaluate the neutralizing antibody response that was generated in non-human primates. And then, of course, we also collaborated with Jim Crow on his, neutralizing antibody discovery pipeline, antibodies that were later licensed to AstraZeneca, as well as, antibodies from Eli Lilly. And this was even at the time before I understood what MTAs were and all that, because I was this eager beaver just waiting, to essentially asking the companies to send me the MTAs. And I think at some point, Ralph very, kindly intervened and told me he's just so eager, but we have to work out the MTAs first with these reagents. So I was more well versed in that later, but
sure.
And then of course that evolved over time. So initially it was just primarily collaborating with companies. Ended up also collaborating with the VRC with the team led by Kizzy Corbett on looking at the neutralizing antibody response of mRNA 1273 in non-human primates. And of course, whenever we looked at the correlations of the pseudovirus neutralizing antibody data that they were generating in our live virus data, it looked really well. So we were very pleased about that. And then eventually, what I sort of did with that was more towards my own program, which was really around, the theme of identifying Achilles heels on not just SARS-CoV-2 and variants, but really around SARS-related bat zoonotic pre-emergent viruses from, either SARS-related viruses and MERS-related viruses. So, we identified a variety of these types of Achilles heels utilizing various types of assays, including these high throughput neutralization assays. And then eventually, I was using those viruses for testing some of our universal vaccine concepts for a wide variety of coronaviruses.
Well, that's great. And, just your story is really inspiring because it does tell you you have an expertise in a particular thing under the right conditions with the right mentorship. It can really lead you to great places. So in our last couple minutes, we want to hear about your current program at Yale, what you're what are you excited about for the future? And then what would be a piece of advice that you would give a trainee aspiring to be a biomedical scientist?
Yeah, those are, great questions. So we are still focusing a little bit on coronavirus immunity. So sort of continuing building on this theme of, universal based approaches to coronaviruses, we're now focusing on, testing some of those, what we think are some of the most promising concepts on airway delivery of various vaccine platforms, to really try to, have better strategies that can reduce viral transmission. So I think that's really a gap right now in our understanding in terms of, fine-tuning strategies that can, I wouldn't say block reduction. I think that's a very high bar and sort of getting at, the unicorn of sterilizing immunity, which I don't know if it exists or not, but more so strategies that can, maybe reduce transmission. So we're kind of focused on that. That's a little bit more applied. And then most of my program is actually revolving around flavivirus pathogenesis and immunity. So, this concept around reverse vaccinology, which we think of as studying antibody responses in people that are very protective or not, that are perhaps disease enhancing, like some antibodies can be against dengue virus, and then understanding broad concepts of immunity against vaccines that work very, very well, like holy grail vaccines like the 17D yellow fever vaccine, which is a one-shot dose that can typically protect for life in most people. And then, trying to understand a totally opposite vaccine, so a Dengvaxia vaccine, which is the only FDA-approved vaccine for dengue, a vaccine that not only temporarily protects, but in fact, in seronegative children, makes them more susceptible to severe disease. So really sort of understanding broad rules of immunity and the exception to those rules, utilizing these types of systems. So sort of a tale of two vaccines is something we're initially focused on. And then, there's other things that we're initially starting to explore around neuroinvasive flavivirus pathogenesis. So not ready for prime time yet, but, sort of exploring new ideas and, trying to utilize, mouse models that we can, readily manipulate to really understand viral pathogenesis and biology.
Well, think. Oh,
did
yes.
you want to? What's your piece of advice?
Yeah.
My piece of advice, I would say be persistent and be patient and enjoy it. So that's one of the things that I think even I have to remind myself from time to time is to try to enjoy the journey just as much as the destination. So that's one thing that, I maybe wish I would have done a little more was to enjoy the journey. Because you can kind of get caught up in the destination that whenever you get there, there will always be a next destination. Right?
Yeah,
So you get to assistant professor, hopefully, right? If things go well, there should be a next destination, right?
Yeah.
You'll be reaching for associate, then full, and then, you'll always be reaching for something. So try to enjoy the ride.
I love that.
Yeah.
Well, congratulations as well on your early career award through AAI, which is the Aspire Award. You're going to be giving your talk tomorrow and receiving that award. And so we're really excited for you for that. And anything else you want to share with us before we end today?
I don't think so.
Okay.
All right. Well, thank you very much.
Yeah. Thank you so much for joining us.
Thank you.
That's a special episode of Immune, an immune booster. If you have questions or comments, please send them to immune@microbe.tv. If you enjoy our work, please consider supporting the science shows of Microbe TV. Go to microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today is from the Yale Institute for Global Health, David Martinez. Cindy Leifer is at Cornell University at Cindy Leifer on X. Steph Langle is at Case Western Reserve University at Stephanie Langle on X. Music on Immune Boosters is by Tatami. Thanks for listening to Immune, a podcast that's infectious.